So Jonathan is also sticking with the ectomycorrhiza theme, and Jonathan's is a masterful genomic scientist. So he's going to share some of his expertise and stories on Pisolithus. So you can take it away.
Perfect. Thank you, David. You can hear me all fine? Great. So thank you very much, David, for the invitation, and thank you all for attending today. I'm coming to you from the beautiful Darug country here in the Hawkesbury in Australia, and I want to talk to you about some of the work that we have been doing within the genomic sphere, as David was saying. But while I'm presenting today, what is represented today is actually going to be about 10 years worth of work, dozens of different authors who I'll put up here and thank right at the beginning, because this is not a one-person job. This has been a heck of a lot of work. So because this is supposed to be the New Zealand Fungal Foray online, I thought I'd talk a little bit about some of the experiences I've had with when people ask me what I do. So usually when I mention the fact that I'm working with fungi, I get this. Oh, you work with mushrooms? That's so cool. Which one? Which then usually leads to these lovely stories that people tell me about, oh, well, do you work with that really cool red mushroom, you know, the one with the white spots? And I'm like, nope, stopping you right there. That's Amanita. Don't work with that. Oh, okay. Well, do you work with those, you know, really pretty tiny ones? So romantic and nope, not those either. So at this point, people are really stumped and they're like, well, what are you working with? And I say, well, I work with this mushroom called Pisolithus. Usually that leads to blank looks. So I say, well, it's more commonly referred to as dog poo fungus, which then leads to really confused looks. And they say, well, why is it referred to as dog poo fungus? So I show them a picture of what I work with, and then they kind of understand. So Pisolithus, well, it might not be the most pretty of all of the different mushroom-forming fungi that you can think of. I think it's pretty cool. And I hope today that you'll be able to get an idea of some of the coolness that you can get out of it when you look at a really, really small scale of genomics and genes. So Camille has really defined a lot of the next few slides beautifully. May I just say I want to travel to Patagonia now. So what we're going to be looking at today, again, is continuing within the mycorrhizal fungal field and sticking with, again, the ectomycorrhizal fungi. So this is an example here of an ectomycorrhizal fungus calling a pine root. So a root would usually be, you know, this lovely brown color, but when it's colonized by a fungus, it gets quite fuzzy. And then you have the fungal hyphae that are extending out into the soils from there, and that's what really is prospecting the nitrogen and other nutrients like Camille was talking about. So the central dogma usually is around this idea that ectomycorrhizal fungi form a symbiotic relationship with the tree because they're looking for a source of carbon. So it's assumed that plants are photosynthesizing so much that they have this extra amount of carbon that is a luxury accumulation they don't need anymore. So they share it with microbes around them in return for a number of different growth limiting nutrients, such as nitrogen. But as Camilla already pointed out as well, you have a lot of other benefits from these mycorrhizal fungi. So you have other macronutrients. So if you look back in the literature into the 80s and 90s, especially with the ectomycorrhizal fungi, people didn't look at nitrogen, they were looking mostly at phosphorus. You have other trace nutrients, you have protection from drought, stress, pathogen protection, etc. So the ecological role of these fungi is actually very broad, and I think we're still just scraping the surface of what they do. What I'm interested in, though, is really this idea of how does an ectomycorrhizal fungus colonize a plant root. So this is a cartoon here where cells in green are a cross-section, or a transverse cross-section of a root. So, you know, as if you were to cut your finger like this, where you have the rhizodermal cells here, cortex all the way to the vasculature. In the second one, you have in light tan here these blobs, which are supposed to represent hyphae, fungal hyphae. And what you can see is they begin during colonization to aggregate on the root surface, and then they begin entering in between root cells. Now, with ectomycorrhizal fungi, these never penetrate root cells unless the root is dying, and instead they stay within the apoplastic space and form what's called a Hardig net within the root. This is where the nutrients are exchanged, like nitrogen, phosphorus, etc. But if you think of the logistics of how the heck this is happening, there are three main areas in my group that we're looking at. And it's the question of, well, it's quite invasive. So how is the defense or immunity in the plant being turned off or at least reduced so that the plant doesn't reject this fungus out of hand? Next, we have this idea that the fungus has to penetrate in between cell walls. It should really be glued quite well to each other. So how is the plant cell wall being rearranged to accommodate that ingrowth? And then finally, that idea of nutrient flow. How is that established? How is that being maintained? So Physolithus is a really great model in the fact that it is found globally and it has a lot of different hosts like those that are seen here. So we have eucalypts, we have manuka or canuka, and we also have pine. So it really has this range of hosts. However, it gets really complicated once we begin looking into the structure of this. And Teresa Labella has been the one who's really been pushing this forward and trying to identify and define for us what a species actually means within this genus. And now I am not a mycologist in general, but when I get with or into the phylogenies with Teresa, I sit there and my head starts to hurt. However, from the work that she's done, what we can see is that there's at least 19 different species that we can find within this genus, but there's evidence of a lot of cryptic speciation as well. Host-wise, there's at least 50 known hosts, and the importance from this is a lot of these hosts, as I showed in my last slide, is that a lot of them are economically important. So in trying to figure out how to grow the hosts better, if we can look at the microbes and understand what the microbes are doing to support that growth, maybe we can come up with some good, sustainable ways of improving our production of these host plants. As I said, they are found globally. Now, Physolithus is found globally, both because in some ways that is where it naturally comes from, but mostly we're seeing an expansion as well due to industry. Because as people are taking pine and bringing it to different countries where it shouldn't be, same with eucalyptus, other bushes, it's just being its fungal symbionts are being moved with it. However, within Australasia, we have an especially high diversity of Physolithus naturally. So it's anticipated this is really where it has evolved. So the work that we've been doing, as I said, for the last 10 years is trying to refine our understanding at the genomic level of how similar these are at the gene level and whole genome chromosome level. Because this is the New Zealand Fungal Foray, I thought I would bring in here the fact that we haven't just looked at Australia for this, but we've actually traveled to different places to try and isolate these fungi. And so in 2016, I had the honor of going with Ian and Teresa to New Zealand. Now, Physolithus in New Zealand is only found in restricted areas as far as we know, and that's basically the thermal areas. And so not only did I get to go to New Zealand, but I got to go to these really cool, beautiful areas. And so these are two of the pictures that we took while we were there. And we were able to get fruiting bodies or mushrooms from under bushes like this, but also like in craters of the moon. You can see here that we have fruiting bodies or mushrooms that are extending right down into these actual thermal vents. And if you've ever been there, you know, high amounts of sulfur, really smelly and really quite hot. So this would indicate not only, can these fungi live in a lot of places globally, but they can withstand a lot of different stresses. So from our time in New Zealand, we were able to get roughly about 100 specimens from the different sites that we went to. And from these, we were able to isolate about 30 pure fungal cultures. And so what this means is that we were able to take a small piece of the mushroom and we were able to grow it in a petri dish and maintain it there. And as you can see from this image, even though the fruiting bodies may look very similar, and this is a mixture of Physolithus albus and Physolithus microcarpus, You can see that when they're grown in culture, their hyphae have very different growth morphologies, colors, etc. So already we can begin to see that these fungi, even though we might define them as one or just maybe two species that are closely related, how they're even just growing is very different. So from New Zealand, we were able to recover these 4 species, and as best as we were able to tell from ITS sequencing. But of interest for us at the time was initially we were trying to take mushrooms that were quite far apart from each other, because we thought, well, any mushrooms that we find side by side under a single tree are likely to be the same species or even the same individual. But for those few places that we were able to get mushrooms that we did take them side by side, we found that they were in some cases very different species. So this would suggest to us again that, you know, they might be a lot more diverse functionally than we might have anticipated, because ecologically, technically, they should only be co-occurring like that if there might be some functional complementation. So beyond those 4 species that we were able to recover from New Zealand, when we combined this with our fungal cultures here from Australia, as well as those that we've received globally, we were able to start this pan-genome project with 9 different species as they are listed here. And the idea was to try and see, okay, if we expand beyond ITS or some of these other loci that we use when we're making these phylogenies like you see here, what can we use to define the genome? So I'll start today with my talk at looking very high level at the genomes, and then I'll use a couple of vignettes to see how we've used these genomes to try and understand how they colonize a host. So the first thing we usually look at within a genomics context is size, because size potentially actually does matter when it comes to genomes. Now, within the Basidiomycota, where you find Piezolepis, the rough genome size is 46 megabases, so this is 46 million base pairs. And within that, you have roughly about 15,000 genes. So this is the average. However, as we all know, fungi are very different. And so there is actually quite a spectrum of sizes. So some examples that we can think of, you have the Champignon de Paris that you would pick up in the local stores. Its genome is 30 megabases with 10,000 genes, fairly average. You have others which are known to, like this one, which is a pathogen of chocolate, of cocoa, and it has roughly the same size of a genome as agaricus, but it actually almost has, again, 2 times the amount of genes. So that's quite a gene-dense genome. And then we have on the other end, the tuber melanosporum, or black truffle, which has a ginormous genome for a fungus, but actually it has very, very few genes. So we would call this a gene poor. And the reason that we are increasingly thinking that we have this diversity within genomes and what's actually potentially driving a lot of the evolution within these genomes is the activity of what we would call transposable elements. Now, transposable elements are a genetic feature that has been known for a long time. They were discovered in the 1940s by Dr. Barbara McClintock when she was studying maize. And she proposed, when she was looking at some natural mutants, that there may be genes or pieces of the DNA that were able to jump around within the genome and cause different mutations. Her work initially was received with a lot of shock and flack, but she was able later on to prove this, and it was also found within bacteria. And what happens with these transposable elements and why they're called jump-in genes is because they do 2 different things. They are of 2 different classes. In one case, it's a piece of DNA that is able to self-replicate itself, so copy itself, and then that copy floats around and then randomly inserts somewhere else in the genome. That's number 2 in the picture here. Number 1 in the picture is also the other types of transposons or transposable elements that self-cut them or cut themselves out of the DNA, again, float around in the nucleus and then reinsert somewhere else. So you can imagine that by their activity, they are able to rearrange a genome on a short time scale, but also for those that are able to copy themselves and then reinsert somewhere else, that increases this genome size without necessarily adding genes. So these transposable elements within the kingdom of fungi have really been, we think, one of the driving forces of leading to the evolution of what we see now. So how does Physolithus actually compare to these, examples I've been giving you? So on average, and what you're looking at in this picture here are these gray bars here to indicate genome size. Physolithus is fairly, average across the board. So it has roughly 40 megabase genomes, so that's fairly average. Gene coding space, it has about 16,000 genes. Again, pretty much bang on when you look across the Basidiomycetes, what you would expect to find. However, the density of those jumping genes, or those transposable elements that I was talking about, is actually really high. So here we have, a list of all the Physolithus genomes that we have sequenced, compared to a lot of other mycorrhizal fungi, as well as brown litter rotters and pathogens. And what you can see here in the TE level, you can see that they account for up to 60% of the genome space is just these non-coding TE genes, versus some of these other genomes which are very poor in transposable elements. So the fact that Physolithus has such a high level of transposable elements leads to the question is, okay, well, if there's so much of these, and if they have been active in the past, is this actually leading to a high amount or more genetic diversity than we might have anticipated? The answer is yes. So when we looked at the genes and compared them between these species, rather than finding almost all of the genes being held consistently between the species, thus a core gene, we actually found that only 13% of the genes that we had sequenced actually were found in every single one of the genomes. Conversely, we found that 63% of the genes that we found were unique to a specific genome. Therefore, Physolithus, even though the fruiting body may look the same, even though you may find some of these species co-occurring, they actually have very, very different gene complements. However, that's at the broad scale of saying, oh, single gene, or sorry, this is the fine scale, to say that a specific gene is only found in one genome, that's looking just at the sequence of the gene. When we look at the function of these different genes, say in sugar metabolism, we actually find that there's a relatively consistent ratio. So that means that within the core gene of those 5,700 genes, we would find roughly 5% of them might be associated to sugar metabolism. Similarly, in the specific genome, so those 26,000 gene clusters, we would again find that roughly 5% of them would be associated with sugar metabolism. However, it gets complicated from there in the fact that even though there might be 5% of both groups that are associated with sugar metabolism, we find that within the core genome, so within the genes that are maintained across all the genomes we've looked at, those genes associated with sugar metabolism are associated with basic sugar construction, glucose, sucrose, etc. Whereas in the specific genome, so those genes that are specific to each species, that's where we find the enzymes responsible for synthesis of symbiotic sugars like trehalose. So there is still some interesting divergence there that we have to look at a little bit more. So some conclusions that we've had from just looking at the genomes is that they are far more diverse than we had anticipated. And this could be potentially the fact of the activity of transposable elements in the past. But this diversity may explain why it is so hard, even using a multi-locus phylogeny, to actually define what an actual species group is. However, for those people like me who are working at trying to understand, the mechanisms that allow an ectomycorrhizal fungus to colonize a host, it's a potential gold mine, right? Because you have such divergence here. You have lots of different genes you can look at and compare and contrast to understand what is the minimal number of genes you need to colonize a host, and how does adding more genes or more diversity to that actually affect different things like host specificity. So as I said, I wanted to look at a few different vignettes of how a fungus may affect or lower the defenses of its host. So I would term this as, how does a fungus manipulate its host to colonize? we can also look at this idea of how does the fungus grow into the root, how does it rearrange plant cell wall, and probably it uses a class of enzymes known as cazymes, which I'll get into. And then how does it provide or maintain that nutrient flow? And this is probably in part due to a source sink kind of gradient. However, this is really scientific speak. I like to think in pictures. And so another way that you can think of this is, how does the fungus hack the security codes, or how does it hack the immunity of the plant? Once it's done that, how does it break and enter? And finally, how does it lock away the loot? How is it getting that sugar from the plant and locking it away to maintain a concentration gradient to keep the sugar coming? So let's look at that. So within fungi, whether they are beneficial microbes or others, we know that a lot of disease-causing microbes or even insects or nematodes use a battery of proteins called effectors. So these are secreted proteins that get into the host or stay near to host cells. And essentially what they do is they reprogram or hack how the cell operates, such that it benefits the colonizing disease microbe, or it allows the aphid to feed, etc. So there's a lot of different ways that you can define what an effector protein is, and you can see them here. But essentially, these are proteins that have evolved within each species of fungi that are secreted and somehow manipulate their host. Within mycorrhizal fungi in general, so far at the level of functional characterizations, there are relatively few examples that have been published, and a lot of them just within the last few years. But I want to kind of focus on how a couple of these are actually operating. So the first one I'll use is an example from an ectomycorrhizal fungi called Leucaria bicolor, and it's a protein called MISP7. So this protein, we have found, actually interrupts plant hormone signaling. So much like humans need hormones to develop and operate well, so do plants. Plants use a lot of their different hormones as part of their signaling that induces or represses immune responses. And so this fungal protein called MISP7 is known to get out of the fungus into the plant cell, where it targets a plant hormone receptor. It binds to that receptor like this in this cartoon, and when it's bound there, what happens is that it affects the ability of that hormone receptor to perceive the plant hormone. Essentially, it stops the ability of that complex to bind to that hormone, or at least that's our current idea. And what this does is it stops downstream plant hormone signaling, and that interruption is what enables root colonization by the mycorrhizal fungus. So in this case, MISP7 is a hacker that is going into the plant cell and really reorganizing how hormone immunity is working. But we're talking about Physolithus today. So it too has different proteins which we've been able to identify what they're doing. Currently, only one of them is published, and that's one called MISP10. And what it does is it affects secondary metabolism within the host. Now, metabolism, much like it says, is it's taking a lot of different building blocks and it's making different components within the plant cell. So this really complex pathway is all centered around this metabolite or compound called adenosylmethionine. And the reason it's important within plants is that it feeds a whole bunch of different pathways. So it feeds the Yang cycle. It affects protein methylation. It produces another hormone called ethylene. And then it also produces or is used to produce polyamines like spermine, spermidine, etc. The reason this pathway is important within understanding how fungi colonize plant hosts is the fact that ethylene, when it's produced in high quantities, inhibits colonization of these fungi. Protein methylation is messy, but it can induce or repress colonization. But polyamines pretty much universally help colonization of mycorrhizal fungi on their hosts. So that's within the plant, just normally. However, when you add MISP10 in, so when Physolithus produces this and it gets into the plant cell, what it does is it affects the enzyme that shunts or ferries adenosylmethionine and makes it or it turns it into these polyamines. And so when that effector MISP10 is present, it's hacking the system, it's altering how an enzyme works, it's causing more adenosylmethionine to go into this polyamine pathway, and it's inducing colonization or promoting it. We think either directly or potentially from removing this building block from a negative pathway like ethylene. So effectors, these are one of the main ways we think that fungi are able to hack immunity within the plant. However, that's just the first step needed before the fungus gets into the plant. How is it actually breaking into the root? And so we think that how the fungus is then able to manipulate the root physically is through an enzyme family known as cazymes. So cazymes are called carbohydrate active enzymes. They are a huge class of enzymes. They do a lot of different things, from breaking down complex cell walls to softening connection between cells through to sugar metabolism. So they do a lot of different things. However, especially within these first two sections, you can imagine some of these enzymes being like the crowbar in that picture that I was showing, in the fact that they can get either soften the connection between those cells so the fungus can get in, or it can actually fully break down the cell wall if a particular fungus needs to feed on the interior part of that cell. And based on this, there have been predictions made before and during a lot of these comparative genomics projects that when you think of a saprotrophic fungus, so this is something that lives off of dead complex plant material that has to break down plant cell walls, you would imagine that it would have a lot of cazymes, versus pathogens, which do a little bit less damage, and then mutualists, which just need to wiggle in between cell walls. You would imagine that there would be decreasing number of cazymes within these genomes. And to date, if we zoom out to a lot of the comparative genomics projects that have been done, this is actually true. So it is found that a lot of these different saprotrophs have a lot fewer cazymes, or sorry, these saprotrophs have a lot more cazymes, and beneficial microbes usually have far fewer. And within those, usually only really gentle cazymes that might enable the fungus to wiggle in between these plant cells. And in 2018, we had our first taste of that, where Feng Zhang was able to prove that one of the cazymes within the Carya bicolor, an ectomycorrhizal fungi, which are pictured here in green, being secreted from fungal hyphae, which are these little blobs. What it does is this cazyme is able to soften the connection between these plant cells. So you can see here in large dark areas, these are the plant cells, and the fungus is able to penetrate in between them, we think because of GH5 softening the connection between them. So how do Physolithus species compare to what has been looked at so far? Like previous, it was found that Physolithus does have a restricted set of cazymes. So again, this is going back to that original list that I had. So as you can see here, there are very few total count of cazymes within the Physolithus genomes, especially when we compare them to saprotrophic fungi and a few pathogens. So that's good. That makes sense that they would have very few cazymes, and those that it retains are actually those softer ones that are just to manipulate connections between cells. However, then it became a bit weird. We found that when we looked at what specific cazymes were retained in Physolithus, there were some really distinct differences. Now, what this image is here is an example of when you look at all of the different cazymes within all the fungi that we look at and just group based on similarity and differences. We found that Physolithus, all the genomes were kind of grouping off onto the side all by themselves. What this means when you look at an image like this is the farther apart dots are, so like Crocyorhizis here, versus some of these out here, that means that they have very different cazyme complements. But color-coded is the lifestyle. So Physolithus is even different than the other ectomycorrhizal fungi that we considered. And the reason for this is because within the cazyme profile of Physolithus, we have this unique amplification of sugar metabolism genes. And this leads me to the very last part of my talk, which is the idea of locking away the root. So how are these fungi actually funneling carbon into themselves? Because, as I said at the very beginning, one of the key aims within symbiotic fungi is to gain that carbon from the host, okay? And while it's a really multifaceted procedure, that involves transporters and synthesis and all this other stuff, one of the only factor I'll look at today is the idea that if the fungus is able to take a really simple sugar like glucose and lock it away into a complex sugar within the fungal hyphae, that creates a concentration gradient whereby glucose will continue flowing into the fungus just because there's very low amounts of glucose left over. So when we were looking at the cazymes within Physolithus, it turns out that there were a lot of these genes or enzymes responsible for turning simple glucose into a really complex storage molecule called trehalose. So we said, oh, great. We've got this idea that, if there's more of these genes within a genome, the automatic assumption is that the fungus will be really great for taking up carbon from its host. That's a great idea, but we thought we'd ground truth that. So again, here's our phylogeny of the different species that we looked at. And we looked at the gene complement, so the number of genes of trehalose biosynthesis genes within 4 of these. So we found that Marmoratus had the highest number of these genes versus Microcarpus, which had the lowest, and then we had a couple in between. So you would assume based on this that Marmoratus would be excellent at synthesizing trehalose, locking it away and getting a lot of carbon from its host, whereas Microcarpus would actually be fairly pedestrian. It wouldn't be able to do that very well. However, what we found when we actually looked at the sugar accumulation was that Marmoratus had, in green here, almost no trehalose accumulation versus Croceorhizis had bucket loads of it, and Microcarpus had kind of somewhere in between. So this would indicate to us that just gene number is not enough to explain this. We then looked at gene expression, and so what this is, these are cut out pieces of heat map. Every single one of these stripes is a different gene, and the color from white to red indicates the extent of the expression. And so what this would say to us, though, is that even though Marmoratus has lots of genes, even though they're expressed quite highly because they're in purple to red, it's still not making very much. So that means that actual genome number of genes isn't always enough to give us or tell us about a gene or what the phenotype will be in the end, nor is transcription by itself. So we really have to combine a lot of different aspects within this to understand the actual final phenotype. So this is the caveat to all of these genotype level looks that we've had. You have to do some characterization at the end to ground truth what you're finding. So some of the mini conclusions from that is that even though Physolithus does have differences in trehalose synthesis enzymes, that doesn't necessarily always translate into actual formation of trehalose. But this is also an interesting finding in the fact that because we found such differences in the ability to capture carbon from its host, normally if you were just doing an ecological survey and you say, oh, well, we have 5 Physolithus species within this one soil type colonizing this one tree, initially you might have assumed, well, they're all doing the same thing to the same level. But actually this is saying to us that the species are different in their ability to recover carbon from their host. So we can't ignore the fact that species actually matters. So in conclusion, just a few of the take-home messages that we have. So Physolithus species have highly specific genomes, far more than we would have anticipated. So this might indicate or ask the question, is the core identity of what a genus is it actually way smaller than we thought it was? Does it only involve just a handful of genes? For Physolithus, it's able to hack the immunity of its host using these secreted proteins, but every single ectomycorrhizal fungus produces dozens, if not hundreds of these. So how is the plant actually coping and interpreting these signals? Physolithus, like other ectomycorrhizal fungi, does have a reduced complement of cazymes. But what will happen if we remove some of these? We haven't tested that yet. Are they actually essential for colonization? And finally, within this idea of how the fungus actually is able to recover carbon from its host, it could be partially driven by the complement of cazymes, But this is still very much in its infancy of our understanding that. So what are some of the regulatory cues of what actually modulates this synthesis? So with that, I'd like to thank the 3 major funding organizations that have funded this. And with the last second I have, I'll just say that within our group, we have 2 postdoc positions opening up. The one on pine at the bottom is currently advertised, and the one on eucalyptus is hopefully going to be advertised in the next couple of weeks. So if you know somebody amazing, or if you yourself want to work in this area, please do contact me. With that, thank you.
Wow, thank you so much. Yep, gosh, what a daunting amount of information there. I guess it's incredible that you just discovered that every one of those species does something in a different way, and you didn't give up. You actually took it further and managed to succeed in actually starting to understand it. And I just couldn't help thinking that once you bring in all the thousands of mycorrhizal fungi that are not even able to be cultured, it really opens the question of what the hell are they all doing individually, not just collectively.
So true, so true.
Gosh. Okay, this is great. So Camille has her hand up, and I'm just going to make you a, oh God, no, I do not want to do that. Make her a co-host. There we go.
Thank you so much, Jonathan, for this very interesting talk. I was wondering if you have looked at the genome of some of the endophytic, especially the wood endophytic fungi, because there is this theory, you know, that they're like in the waiting room for becoming mycorrhiza. So I was wondering if you have looked at the genome of some of them, and if you found some kind of like intermediate patterns, for example, for some of these genes. So.
Yeah, that's a great question. No, I have not. Even though I love looking at these genomes and doing comparative genomics, I really am a nuts and bolts person. So I like really understanding. So I get, you know, I love the forest, but I like looking at the, you know, single trees. So no, I do work with a number of people who like looking in those areas. The problem that we're running into, though, is just like we have, you know, at this genus level, the vast degree of differences, even within a species, not that I wanted to say this, but even within a species, we're having huge genetic diversity, right? And so what does a waiting room look like? What does an intermediate look like? It would be really curious to look into that with some of these endophytes, right? Or are some of these endophytes actually, another theory that we would look at too is this idea that because you have all of these mycorrhizal fungi that are essentially hacking the system, right? Are they just taking advantage of the fact that, oh, look, this root has slightly lowered immunity, and they just kind of colonize based on that, right? So are they an intermediate, or are they just taking advantage of a lot of these other fungi doing the work for them? But no, fascinating question. I myself have not looked at that.
Okay, yeah, I was thinking about this because as one of the previous speakers show, they are very easy to culture sometimes, so it should not be too difficult to get the genomes, I guess.
Yeah, we're doing a lot of work now with the pine industry, much like is being done in New Zealand as well, and doing a lot of these sequencing of even just individual root tips of these mycorrhizal root tips and all of the diversity of fungi that are there. And trying to culture it out of them, I get really frustrated, right? I want the ectomycorrhizal fungi. But now more and more, it's like, okay, well, how many of these others are codependent or, you know, supporting the efforts of the mycorrhizal fungi, et cetera? It's a fascinating area, especially for people like me who typically work in petri dishes, right? I mean, it's only since coming to Australia that I'm working more and more with the ecological implications of this, you know, like working with David and working with Teresa, and it's just like, mind-blowing. My brain is really too simple, honestly, to try and understand everything all at once, but it's really cool. So I think it'll be great, you know, as these endophytes are sequenced and we begin to understand how they may be working together, will be really cool.
Thank you.